How does reproductive cloning use embryonic stem cells ethically? Have you ever wondered how reproductive cloning interacts with the world of embryonic stem cells? This topic raises many ethical questions that are important to understand. Reproductive cloning is the process of creating a genetically identical organism. This is done by transferring the nucleus of a somatic cell into an egg cell. This method is known as somatic cell nuclear transfer. Embryonic stem cells are derived from early-stage embryos, often created through this cloning process. The ethical considerations surrounding the use of these stem cells are shaped by several key medical ethics principles. These include respect for human dignity, informed consent, beneficence, non-maleficence, and justice. First, let's talk about respect for human dignity. This principle requires careful thought about the moral status of the embryo. Since embryonic stem cells come from embryos that are destroyed during the derivation process, there are ethical concerns about ending potential human life. Some argue that embryos created specifically for stem cell use are different from those intended for reproduction. Others suggest limiting the creation and destruction of embryos by using only surplus embryos from fertility treatments or following strict regulations, like the 14-day rule, which restricts embryo research beyond a certain developmental stage. Next, informed consent plays a crucial role. All donors of somatic cells, egg cells, and surrogate mothers must give voluntary and informed consent. They need to understand the purposes and risks of cloning and stem cell research. However, the cloned individual cannot consent to their own creation, which raises ethical debates about autonomy and the rights of the clone. Beneficence and non-maleficence are also important principles. These requiring that reproductive cloning and the use of embryonic stem cells aim to maximize benefits while minimizing harm. Currently, reproductive cloning carries significant risks due to genetic abnormalities and developmental defects seen in animal clones. This raises safety and welfare concerns. Ethical use means that cloning should not proceed until these risks are significantly reduced to avoid harm to the clone. Justice is another principle that focuses on equitable access and fair distribution of the benefits and burdens of cloning technologies. Ethical frameworks call for regulations to prevent exploitation, misuse, or discrimination related to cloning practices. In practical terms, many medical ethics guidelines and laws prohibit reproductive cloning due to safety concerns and moral objections. However, therapeutic cloning, which uses somatic cell nuclear transfer to create embryos for stem cell derivation aimed at treating diseases, is allowed under strict oversight. This distinction helps maintain ethical standards by separating reproductive intentions from research aimed at medical benefits. In summary, the ethical use of embryonic stem cells in reproductive cloning relies on respecting human dignity, ensuring informed consent, prioritizing safety to prevent harm, and regulating access to maintain justice. These principles guide the responsible development and application of cloning technologies in healthcare today.